Why do some animals live so much longer than others? And what is it that causes the variation in the aging process? One theory suggests that somatic mutations, changes that accrue in our bodies over time, could be part of the answer.
So there's good reason to suspect that somatic mutations that are altering the DNA could be having deleterious effects on cellular function that could lead to breakdown and kind of consequences that we see in aging.
But this theory doesn't necessarily align with what we see in the animal kingdom. For example, somatic mutations are known to cause cancers, which can in turn reduce lifespan, and so it would stand to reason that larger animals with more cells to mutate might have a higher chance of developing cancer somewhere in their body and thus have lower lifespans. But in fact, the opposite is often true. With a few notable exceptions, larger animals often have far lower rates of cancer than smaller ones, a phenomenon known as Peto's paradox.
From everything we know about somatic mutations and cancer, we would expect that the more cells you have, the higher your chance of cancer, because you just have more cells where you could be unlucky and accumulate somatic mutations.
What's more, some of the longest lived animals in the world are also the largest, despite having so many cells which could be deteriorating through mutations. If somatic mutations are linked to aging, then something else must be going on. And now, a team of researchers have delved into the somatic mutation rates of a range of mammals for the first time, in the hopes of finally unravelling some of these mysteries. Somatic mutations can be caused by lots of things, like errors in DNA replication or exposure to mutagens like ultraviolet radiation. Despite being very common, their effects are still not fully understood.
The most well-known consequence that's very well established is cancer. But what's less well understood and still debated is whether somatic mutations could be playing other roles. Crucially, for example, what is the role of somatic mutation in aging? Because we know that as we age, we're accumulating somatic mutations, but kind of the key question in the field is, are these a consequence of aging or a cause of aging?
Alex and his colleagues wanted to measure the rate these somatic mutations were acquired, not just in humans, but across as wide a variety of mammals as possible. So they set about collecting tissue samples.
So we work with a variety of partners, including the London Zoo, so ZSL Institute, and the CSIP, which is the Cetacean Stranding Investigation Program, who I'd never heard of before, but they're essentially like CSI for cetaceans or whales. So we work with partners like that and different zoos to collect tissue samples opportunistically from when animals had died from other causes. And so we wanted to get a tissue type that we could compare across mammals, because we know from human studies that different cell types in the body have different mutation rates. And so to compare across species, we wanted to pick one cell type, and we settled on intestinal crypts, which are these small kind of crypt-like structures that lie in the intestine. And so basically by sequencing these intestinal crypts from 16 different mammalian species and 56 different individuals, and knowing the age of those animals when they died, we could count and say, okay, in this harbor porpoise that's X years old, there are this many mutations, while in this mouse, that's maybe a few months old, we count this many number of mutations, and then we can figure out the mutation rate across these different species and see to what extent they are similar or to what extent they vary.
Their results allowed them to investigate whether mutation rates can help explain Peto's paradox, the curious observation that larger animals don't appear to develop more cancers despite having more cells to mutate. And it turns out that size is actually a fairly poor predictor of the rate of somatic mutation. For instance, cows, giraffes and horses weigh much more than an average human, but their somatic mutation rates don't follow the same pattern. It's clear that having more cells doesn't mean you have a higher chance of developing cancer, but the weak correlation between body mass and somatic mutation rates across species suggests that lower mutation rates are not behind this, and that large animals like elephants must have another trick for lowering their cancer risk. However, there was one factor which seemed to correlate strongly with somatic mutations, a factor that is very relevant to aging: lifespan.
By far, for us, the most striking finding was we found this strong inverse correlation between lifespan and somatic mutation rate.
For example, the naked mole rat is about 23,000 times smaller than a giraffe, but they both live for around 20 to 30 years. And this new research presents an explanation for such similar lifespans in such different creatures. They have nearly identical somatic mutation rates.
And so what that tells us is that somatic mutation rate is evolutionarily constrained. So there's some reason that evolution is shaping somatic mutation rate to be similar at the end of lifespan across species. And this is consistent with the somatic mutational theory of aging, that somatic mutations play a causative role in the aging process.
Across the animals tested, somatic mutation rates proved a reliable predictor of lifespan, and that provides strong evidence for the theory that somatic mutations are a cause of aging, not a result of it. But the team isn't finished yet. They want to expand their study as far and wide across the tree of life as possible.
From evolutionary theory and what we're seeing in mammals, species with longer lifespans seem to have lower mutation rates. So if we can start to understand how species with even longer lifespans than humans are maintaining their genome integrity and lowering their mutation rate, there's a potential to kind of learn from the solutions that nature and evolution have already found to potentially find ways to lower the somatic mutation rate. Whether that will one day be possible, I don't know, but in principle, if one could lower the somatic mutation rate, that could be a very effective way to do things like lower cancer risk or slow the aging process. But so that's kind of the moonshot, far future thinking of how this kind of research could potentially be relevant.